Hello, everyone. Welcome back to the GEP Pathways walkthrough. This is a continuation of part 2.1 as we begin to perform a blast search of D. melanogaster proteins against the target species genome. Recall in part 2.1, we retrieved the protein sequence for Rheb-PA in Drosophila melanogaster. Now we need to perform a blast search of Rheb-PA against the entire D. yakuba genome assembly. To do this, we will be using the basic local alignment search tool, also known as BLAST, to find regions of local similarity between nucleotides or protein sequences by comparing the nucleotides or protein sequences to sequence databases or to an individual nucleotide or protein sequence. What this will do is this will calculate statistical significance of each match, and ultimately they focus on a few key objects. First and foremost will be our expected value, otherwise known as our e-value, which describes the expected number of blast hits with this alignment score or better due to the chance of the lower the e-value. Simply put, the closer it is to zero, the more significant the match. And our percent identity, known as identities, describes how similar the query sequence is to the database or subject sequence. For example, how many characters in each sequence is identical. Now, the higher the percent identity, i.e., the closer it is to 100, the more significant the match. Now, for the basic local alignment search tool, there are five traditional BLAST programs. There is blasn, which searches nucleotide databases using a nucleotide query. There is blastp, which searches protein databases using a protein query. A blastx program, which searches protein databases using a translated nucleotide query. There's a tblastn program, which searches translated nucleotide databases using a protein query. And last but not least, tblastx program, which searches translated nucleotide databases using translated nucleotide queries. We can see on the table that's currently pulled up on the video, this table which shows BLAST programs, the query types, which is the sequence to match, along with the database, otherwise known as the subject, the function, and common use cases across. Now that we have a better understanding of BLAST, since we're looking for an orthologous region of Rheb in D. yakuba, we want BLAST to search the entire genome of D. yakuba to identify regions of local similarities with the protein sequence of Rheb in Drosophila melanogaster we obtained in part 2.1. In other words, we need to blast our Rheb protein sequence from D. melanogaster against the entire genome of D. yakuba to narrow down the possible regions where the Rheb ortholog could be in D. yakuba. For this blast search, we will be using tblastn to search the translated nucleotide database of our target species, D. yakuba, using the protein sequence of Rheb in Drosophila melanogaster as our query. As you can see, with the table removed now, this was that sequence we copied from before. We will be using this in our search. Now, ultimately, there's going to be several things we can think of. First of, the query, which is the sequence we are looking to match. I always like to say the query is like our question, where we want to say, hey, I'm looking for this particular thing, or I'm looking for similarities to this thing. Across our database or subject, which is the collection of sequences we are searching for matches, BLAST will translate the entire genome of D. yakuba before searching for a match to the Rheb-PA sequence for D. melanogaster. But how are we going to do that? Well, to do this, we need to navigate to the Project Pathways Genome Assemblies. Notice how on my other tab, I am already on the Pathways project page. Can be found by simply navigating to Research and Pathways. Once we're here, simply scroll down and go over to the Pathways Project Genome Assemblies. Here, we'll be able to see a complete list of all of the genome browsers that we are utilizing for the Pathways project and their NCBI BLAST link. What we can do is simply search for D. yakuba. As you can see, we could see D. yakuba does show our genome browser, which is our August 2021 Princeton Prin Diac Thai 18E2. Instead of clicking on the genome browser, we're going to go to Genome Blast. Once we're underneath Genome Blast, note, just like we had mentioned before, we'll be starting with the tblastn search underneath the query sequence, we'll be pasting the sequence we had gathered from the protein translation directly from Rheb in Drosophila melanogaster. Here, we do not need to choose a file, nor do we need to provide a query subrange, as we have a relatively small sequence we're searching against. Once this is finished, simply select Show Results in New Window and hit the BLAST button. Now, just for your information, the National Center for Biotechnological Information, otherwise known as NCBI, periodically updates the genome assemblies used for BLAST searches that can cause bookkeeping issues when they occur in the middle of a semester. Using the Genome BLAST link on the Pathways Project Genome Assemblies page ensures students and faculty annotators are navigating to the correct genome assembly database when performing their BLAST search against the entire genome of their target species. However, this circumventilation will only be needed in Part 2 of the Pathways Project protocol. In Part 3 and 5, we will navigate directly to the NCBI BLAST homepage. So, when performing a BLAST search, we may actually return a number of matches, often referred to as hits. For regions of local similarities between our query sequences and databases, however, each hit is not necessarily statistically significant. BLAST provides statistical scores to help us determine which alignment between the two sequences are statistically significant and which are simply spurious, which means they likely occurred by chance alone and are therefore not evidence of real biological conservation. If BLAST returns multiple good hits, which is more than one hit with a low e-value and a high secure sequence identity, we will need to investigate them all further to determine the most likely ortholog. Now, our tblastn search found multiple regions, in this case 5, within the translated D. yakuba genome that shows similarities with the protein sequence Rheb in D. melanogaster. Notice here, especially when we're looking at these general good hits, we are looking for a low e-value and a high percent identity. It is remarkably clear, as we could take a look at each individual blast, which will have the best e-value. We have 2e to the negative 78, 7e to the negative 37, and even things like 3e to the negative 16. We immediately know, as this is sorted in terms of e-value, that our first description here will actually be our best for now. If we look at our percent identity, we can also see that the top result does have the highest percent identity, with its next counterpart only covering less than half, 43.75% of that content. Since we do have the sequence identity of 97.14 and an e-value of 2e to the 78, the second hit doesn't look to be like what we should be looking for, but we want to keep it in mind. FYI, similar to how humans have unique fingerprints that can be used to identify them at crime scenes, each nucleotide and amino acid sequence is assigned a unique accession number to allow scientists to identify it from millions of other sequences stored in the database. Now that we've been able to locate what we need, we can simply click on our possible ortholog, and this will show us and prepare us for part 2.3.